Okay, are there any questions? Can somebody confirm that you're hearing me?
We can hear you.
Great. All right, last Tuesday I sent out the unknown number, and you guys got your bacteria and you started growing them, and you did the gelatin test. And if you remember, you started the gelatin test on Tuesday, and you started the gram stain on Sunday, but you didn't finish it until today. And the the PR lactose, you started that on Monday. And today is Tuesday, so you'll get that result. The reason why you're starting on those days is so that I can get you the results all at one time. But you started the projects on different days, and so it is following dichotomous logic, meaning you're not running 3 tests at the same time and getting 3 results at the same time. I'm just designing it so you will get the results all at one time. To ease my implementation, I will send 1 email instead of 3, which is just a little bit easier. But the whole idea is, instead of sending out 60 emails, I'm sending out 1. Any questions about that project? So when are you guys getting your results? For the first 3 tests?
Today.
Yeah, today. It'll be sometime after the lab. It'll probably be late tonight, because after the lab, I'd like to go on a walk, and that'll take at least a half an hour before I get back. And then I'll try and send that information out to you. So look for that email. That will be the last email you're getting on the project. Everything else that you do on this project should be in your unknown notes page. Let's see if I've got that here. Can you see the screen?
Yes.
Thank you. So if you come down to week 5, the unknown notes page will be under week 5 lab. The unknown notes page is here. From here on, everything you do will be on these pages. Everything I send to you will be on the comment section, which you can't see it right here, but there's a comment section right here. And everything I do will be on a comment. Please do not make a comment. If you want to make a comment, put it in your document and then upload the document. Okay? The comments are only for me. If you start putting comments down, I'm going to tell you I'm going to delete those comments, so you need to move them. All right, any questions? Anyways, that's the unknown notes project. We will be working on that hopefully later tonight. It tells you up here in your unknown notes, record your daily activities, especially your test results requests here. I'll provide test results here as well. Record your results and conclusions here, and list the species under consideration. Everything is dated, so there's no hope on that. I should change that while I'm doing this. That was the very first time I put that up there, and I said, hopefully everything is dated, and I've done it so many times, I know everything will be dated automatically. So I'm going to delete that. All right, any questions about this project? You have from now until the end of the term, or close to the end of the term, to work on this project. It must be turned in, the lab report, Saturday, August 13th. Do not wait until the end to start the unknown project, because this project will take time, which is why I'm trying to get you started, in a sense, last Tuesday, and certainly by today. Why I'm trying to get you started is this project will take time. And if you run out of time, then you run out of time. You can't identify your unknown species. And that's not my problem. I got you started now. If you have any questions about the project, ask me or email me, okay? Don't wait till the end and then ask. All right, let's go to the syllabus. Today in the lab, we're covering lab 7, throat and urine cultures. On Thursday, we'll do lab 8. After I'm done talking about lab 7, I'll stay here for about an hour. Depending on when I finish, I'll stay on for an hour to answer questions. And when the last student logs off, I will log off. So if you have a question, ask me early on, not late on, because I might not be here if all the students log off. All right, let's move on. Lab 7, throat and urine cultures. You know, this looks like it might be the wrong one. Get out of here. Yeah, that's the right one. There's some reading for you to do before you do the lab. We have some video clips. Unfortunately, we do not have a virtual lab. It had to be removed because it no longer works. Your learning objectives: explain how to properly collect throat and urine specimens for routine culture. Be able to list common bacterial pathogens isolated from the throat and urine cultures. Know the different culture media used to cultivate these pathogens. Recognize which tests are used to identify throat and urine pathogens, and understand the principles and how to interpret the tests: catalase, coagulase, and oxidase. We will briefly talk about the XV test, but I don't expect you to understand it and interpret it if I would give you a quiz question on it. Once specimens are received in the lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated for throat and urine specimens. Below is a table of an overview of the culture media used to cultivate common bacterial pathogens isolated from the throat and the urine. For the throat, there's only 2 culture media we use: blood agar, the main one, and then chocolate agar. Streptococcus pyogenes is the main bacterial pathogen isolated from the throat, and it grows on blood agar. However, in children or babies, Haemophilus influenzae can also be isolated, and it does not grow on blood agar. It has to be grown on chocolate agar. For the urine, we have 3 different culture media we grow isolates obtained from the urine. And the first is blood agar again, and then MacConkey's agar or eosine methylene blue, also called EMB agar. Usually you grow either MacConkey's, or you grow the growth usually on MacConkey's or EMB, not both, because both of these, MacConkey's agar and EMB agar, are selective media, and they grow gram-negative bacteria. So you usually only grow 1. Gram-negative bacteria are the most commonly isolated bacterial pathogens from the urine, especially species of Enterobacteriaceae. It's the family that E. coli is in. And actually, E. coli is the #1 bacterial pathogen found in the urine. But there are other gram-negatives like Pseudomonas, and those grow well in MacConkey's and E and B. However, there are some gram-positives also isolated from the urine, such as Streptococcus species, Enterococcus species, and Staphylococcus species. And they will not grow on MacConkey's or E and B, but they will grow on blood agar. So usually when setting up urine cultures, we do blood agar and we do either MacConkey's agar or E and B. Any question about any of that? All right. Sore throats may be caused by many things, viral or bacteria. If you see white patches on inflamed tonsils, it is indicated of a bacterial infection. The most common bacterial pathogen is Streptococcus pyogenes, which I may have pronounced that wrong. I think I said Streptococcus pylori. That's pyogenes in the lecture. Sorry about that if I got it wrong. I have to go back and check. It has a couple of names. Group A, Streptococcus, is another name. I always give it by its species name, Streptococcus pyogenes. Tonsils, white patches, which are indicative of bacterial infection. If it's just red, swollen tonsils, it's probably a viral infection. Strep throat caused by Streptococcus pyogenes may be tested by swabbing the back of the throat or the tonsils. The lecture just finished. That's good. My computer will now speed up. So where the redness or the white patches are, when doing a throat culture, you touch these areas or this area. You do not touch the side of the mouth and the tongue. Patients that test positive for Streptococcus pyogenes are placed on antibiotic therapy. The reason being that humans are not very good at throwing off Streptococcus pyogenes. It has a glycocalyx, specifically called a capsule, that tends to protect it, and so it's hard to throw that one off. Streptococcus pyogenes is fastidious, so the culture media must be supplemented with 5% Sheep's blood cell to get it to grow. The red blood cells supply the nutrients that are required by Streptococcus pyogenes in order to grow. The plate is then examined for any small clear colonies surrounded by a complete zone of hemolysis. Streptococcus pyogenes has small white or clear colonies which have complete zones of hemolysis around them. Only 2 biochemical tests are required for definitive identification of Streptococcus pyogenes. So after you have your blood agar plate showing beta hemolysis, you then do the catalase test and the basotrace and susceptibility test. And depending on the test results, it will be Streptococcus pyogenes. So there's a hemolytic colonies, could be Streptococcus pyogenes. Catalase is an enzyme produced by bacteria to break down hydrogen peroxide. So we have hydrogen peroxide and the enzyme catalase. It breaks it down into water and oxygen. You'll see bubbling when the oxygen is created, and that means that catalase is present. If the bacteria are positive for catalase production, Oxygen is observed. Streptococcus and Enterococcus species test negative for catalase production, no bubbles. Many other bacteria are catalase positive. I can't tell you a Streptococcus species because none of them are that, but Staphylococcus, like Staphylococcus aureus, is positive for catalase. You then do the bacitracin susceptibility testing. Bacitracin is an antibiotic. You put the bacteria, you streak it all over the plate, get it to grow kind of in a lawn or close to a lawn. And then you put down, after you put the bacteria down, you put down the disc containing bacitracin. If the bacteria are sensitive to bacitracin, you have to measure the diameter and determine that it's large enough. I think it's greater than 14 millimeters. Seeing it here. Anyways, that's Streptococcus pyogenes. It's sensitive to bacitracin. Other Streptococcus and other hemolytic bacteria that are catalase negative will not be sensitive to bacitracin, then you'll see no zone of inhibition or a zone of inhibition below a certain size. I'm surprised that's not on here. It's got to be in here someplace. All right, another organism that can be clinically isolated from the throat is Haemophilus influenzae, another gram-negative coccobacillus. It's actually a misnomer because it doesn't cause the flu, but Louis Pasteur thought it did when he isolated it from a flu patient. But in reality, it causes sore throats in small children, especially babies and young children. And it's also a common cause of ear and eye and sinus infections in babies and young children. This is a fastidious, tiny gram-negative coccobacillus that requires growth factors to grow. And the blood agar will not supply the growth factors, but chocolate agar will. If you remember, chocolate agar is the same as blood agar. It's just the blood is added when the agar is still steaming hot, and that lyses the blood and turns the agar to a chocolate color. And there we have Haemophilus influenzae growing on it. The curious thing is that you can sometimes get Haemophilus influenzae to grow on a blood agar plate if you have Staphylococcus aureus growing near the Haemophilus influenzae. What happens is you streak Staphylococcus aureus this way, and then you streak Haemophilus influenzae this way, and you'll notice there's no Haemophilus influenzae growing here, but it is growing in this region near the Staphylococcus aureus. What's happening is Staphylococcus aureus is lysing the red blood cells, and then that's supplying the nutrients for Haemophilus influenzae to grow in this region. Beyond this region up here, there's no lysed red blood cell, and so there's no nutrients from the red blood cells, and so Haemophilus influenzae is not growing. Any question about any of that? in this lab and it asks you, how do you grow Haemophilus influenzae? Just say you grow it on chocolate agar. To try and describe this would be fairly involved. All right, urine cultures. Urine cultures are used to diagnose urinary tract infections or UTIs. Bacteria can travel up the urethra to the bladder. Females are more proposed to developing a UTI than males simply because they have a shorter urethra. To sample the urine, you need to clean the genital area prior to collection and then catch the urine midstream. It's called the clean catch midstream, collecting only the middle portion of the urine stream. If you collect the beginning portion of the urine, you'll get a lot of bacteria from the urethra, which we don't want. We're trying to determine if the bladder has bacteria. And that's why we clean catch midstream, to try and get more of the bacteria from the bladder. This reduces the cultivation of transient organisms from the urethra, although they'll still be there, it does reduce it. Another way is to catheterize the patient and get the urine that way. Patients don't usually like that. Put the catheter into the bladder and then sample it. This is not perfect because you're going through the urethra before you go to the bladder, and that could introduce bacteria from the urethra into the bladder. And then the sampling may pick that up. The best way to do it without any contamination, which patients don't particularly like, is suprapubic aspiration. You put a needle through the abdomen into the bladder, and then you sample the bladder directly that way. And that, of course, is somewhat invasive. Patients don't usually like getting a needle stuck in them. So those are the three ways to do it: clean catch midstream, catheterization, or suprapubic aspiration. Any question about any of that? Why is that over here? All right. Once the urine specimen is obtained in the lab, they put 1/1000th of a milliliter of urine onto a non-selective plate, such as blood agar, using a special inoculation loop that measures 1/1000th of a milliliter. Here it is here. That's 1/1000th of a millimeter. If you have the urine in that loop, and then you just spread the urine all over the blood agar plate. Usually, besides spreading the urine on a blood agar plate, you also spread the urine on a McConkey's or an EMB plate. The reason why we use McConkey's or EMB is that gram negatives tend to be most or the majority of UTIs, and they will grow on a McConkey's or an EMB. They will grow on a blood agar plate, but this will grow many other organisms, gram-positive organisms. And we also grow the urine specimen on a blood agar plate in case the patient has a gram-positive pathogen, like Staphylococcus aureus. It will not grow on McConkey's or EMB. And there we're saying coliforms, particularly E. coli, are the most common pathogen in a urine culture. Any question about any of that? Coliform is just a bacteria that grows in the colon. All right, the interpretation for a positive urine culture is multifaceted. There are different factors that need to be considered. The type of collection, like clean catch midstream, is avoided collection versus catheterization. The clean catch midstream will have more bacterial contamination from the urethra than the catheterization. The gender of the patient is important too. Females have more bacteria from the urethra than males. The number of colonies per mil per urine is a factor. The larger number of bacteria you have per mil, the more likely it is the patient has a UTI. And of course, you will always get colonies because the urethra has bacteria in it. Well, you won't get colonies, contamination, if you use the suprapubic aspiration and do it correctly. The presence of polymicrobial growth. If multiple organisms are isolated, it may indicate a poor collection technique, because usually the patient will have one bacterial pathogen, and it shouldn't be polymicrobial. Any question about any of that? All right. There are a few tests we run after we have a bacterial culture growing. The first is the coagulase test. Coagulase test for the production of the enzyme coagulase, which can be used to differentiate Staphylococcus aureus from other Staphylococcus species. Staphylococcus aureus has coagulase. Other Staphylococcus species are coagulase negative. Coagulase does contribute to the pathogenicity of Staphylococcus aureus. What coagulase does is it takes fibrinogen in the blood or the plasma in the presence of coagulase, and then it clots the fibrinogen, converts it into fibrin, which then starts clotting the region. This protects the bacteria by forming a clot around it. And then the immune system is harder to go in and kill off the bacteria. So it protects the bacteria from phagocytosis, being eaten by the immune system cells because of the clot. And what Staphylococcus aureus does, when it's low in number, it puts out coagulase and digests the clot. And then when Staphylococcus aureus is large in number, a large number of them, they then digest the clot and then are carried by the blood to other locations of the body to spread the infection. All right, we have the rapid slide coagulase test where you put the urine specimen in the slide in the coagulase test. And if it doesn't produce coagulase, there's no clumping. And if the bacteria does produce coagulase, the bacteria will clump together. More likely, I will give you results to the tube test, where you have plasma in the tube and you put in the bacteria. And if the plasma clots, you'll see a clot like this. That would be coagulase positive. And if the plasma doesn't clot, it'll stay a liquid. And this is coagulase negative. Any question about any of that? All right, the most common bacteria isolated from urine cultures is E. coli of the family Enterobacteriaceae. And we'll discuss that family further in Lab 9. There are other gram-negative bacteria that can cause a UTI, and that includes members of the Pseudomonas family, particularly Pseudomonas aeruginosa. How we can separate the Pseudomonas from the family Enterobacteriaceae is that Pseudomonaceae, or Pseudomonas, is containing the enzyme cytochrome C oxidase. And so Pseudomonas aeruginosa has cytochrome C oxidase, and so it'll be positive in the oxidase test. And the members of the Enterobacteriaceae family do not have cytochrome oxidase C, so they're negative in the oxidase test. Any question about that? Pseudomonas and Enterobacteriaceae are both gram-negative bacillus. They have a few differences though. They're both small, but Pseudomonas is aerobic. Enterobacteriaceae is a facultative anaerobe. And then Enterobacteriaceae can ferment glucose, and Pseudomonas, all members of the family, do not ferment glucose. But the easiest way to separate them is the oxidase test. Here we're seeing oxidase positive, it'll be a blue or a purple color, and then oxidase negative, which we don't have shown here, would be no color change, no purple in particular. All right, so that was the summary of what we do to test throat cultures and urine cultures. I wouldn't take a look at the references, but you should take a look at the laboratory 7 exercise. We have some videos for you to do. You're not going to physically performing any of these activities, but you will be held responsible for knowing how to perform them. For the catalase test, we have a clip for you to watch. For the coagulase test, we have another clip. And for the oxidase test, we have a third clip for you to watch. And then we go to the worksheet or the exercises. So answer the questions. What converts fibrinogen into fibrin? And then name an organism that tests positive for this enzyme. So there's 2 parts to this question. 2, what detects the presence of cytochrome C protein in the electron transport system? And then the second part, name 2 organisms that test positive for this enzyme. And the video clip tells you, I think the lab told you, well, it might have only told you 1 organism, the lab manual. All right, 3, what hydrolyzes hydrogen peroxide into non-toxic products, water and oxygen? And then name 2 organisms that test negative for this enzyme. 4, which rapid test is used to differentiate between the following: Staphylococcus and Streptococcus, B, Staphylococcus aureus and Staphylococcus other species, C, Enterobacteriaceae like E. coli and the Pseudomonaceae. 5, concerning specimen collection for routine throat culture, what areas of the throat should be swabbed and what areas of the mouth should be avoided. 6, what is the most common cause of bacterial pharyngitis? You must mention a species, okay? So don't say simply bacterial infection. It's of course a bacterial infection. We're saying bacterial pharyngitis. Now name the species. What test is used? 7, The test is used by microbiologists to differentiate between beta hemolytic group A Streptococcus and other beta hemolytic Streptococcus species isolated in throat cultures. And answer 8 and B only state the two non-selective agar plates that are inoculated when growing the throat culture of a child. And B, what organisms can be isolated on CAP, but not on BAP. CAP being chocolate agar plate, BAP being blood agar plate. C, you can skip because you can't answer this unless you do research online. So you can skip that. That's why I have it crossed out. 9, there's 3 methods that are used to collect urine for routine culture. 10, there's 3 reasons why urine specimens should be rejected for culture. 11, how much urine is placed on the surface of the blood agar plate. 12, who's the most common bacteria isolated from patients with cystitis. Cystitis is a patient who has a urinary tract infection. 13, identify the Gram reaction and cell morphology. You need to give me the Gram stain, Gram reaction, and the shape of these cells. Okay, Staphylococcus saprophyticus, Escherichia coli, Pseudomonas aeruginosa, Streptococcus pyogenes, Haemophilus influenzae. And you can get this from the lab manual, or you can go online. Okay? All right, any questions? All right, if not, I'll be here until 8:40 to answer any questions and help you with the lab. But when the last student logs out, I am going to log out as well. And I'm going to be walking in and out. So if I'm not here, you can't see me. Just wait a few minutes, I'll show up. I'm going to make my dinner, which will just be a sandwich, and then bring it back here. And I'll mute my microphone so you don't hear me eating. All right, any questions? If not, go ahead and get started. I will be here until 8:40 to help you. Come on.